Good afternoon, everyone, and thank you for being here to attend my talk, as opposed to being out there in the nice Okinawa weather today. So my name is Chern Han, and I'm from the National University of Singapore. Today, I will share our work on batch normalization of single-cell sequencing data using our method called MapBatch. Okay, so first off, some motivation. So many diseases involve rare cell populations. So these are cell populations present in very small numbers and can only be found in subsets of patients in a study. So for example, only in certain subtypes of a disease, maybe only in an advanced disease stage, or maybe it can only be found after a certain treatment. So some examples are cancer stem cells, certain immune cell subsets, and so on. Now, single-cell data can potentially uncover such rare cell populations, but this is actually very dependent on the data processing, in particular on batch normalization. Batch normalization is the process to integrate data from multiple experiments so that you can analyze multiple samples together. There are popular methods like Surat and Harmony that do batch normalization, and they perform very well in mixing batches together. But they can actually behave too aggressively. They can reduce the biological signal and obscure certain cell populations. So here's an example. Okay, sorry, let me turn on my laser pointer. So these rare cell populations that are colored here, they can be seen when looking at single sample. But once we integrate this sample with other samples and we perform batch normalization, this cell population becomes obscured. So many potentially interesting biological findings may become lost due to this kind of aggressive batch normalization. So we designed MapBatch, which performs conservative batch normalization. So just enough normalization to enable downstream analysis while trying to maintain as much biological signal as possible. So in fact, we found that for most downstream analysis, it actually doesn't matter much if some batch effect remains. So how does MapBatch actually work? MapBatch is based on autoencoders. So these are neural networks that can project high-dimensional data into a lower dimensional space. Autoencoders are frequently used nowadays in tasks like image recognition. In MapBatch, an autoencoder is trained on one single batch of data. So let me illustrate how this works. So in this data, there are two batches, batch 1 and 2, and they contain the same cell types populations. Now notice that batch 2 is slightly offset from batch 1, and this represents the batch effect differences between the batches. Now we train the autoencoder on batch 1. And what this does is it learns to project the data onto the lower dimensional gene expression structure of batch 1. This is the so-called biological subspace of this batch. And we call this S, represented by this blue curve. Now, when both batches are projected onto S, it looks like this. And unsurprisingly, the three cell populations are still well separated, and the batch effect is very much reduced. Now remember that this is just using one single batch of data for training, and subsequently you can use that autoencoder to normalize as many batches as you want. Now what happens when instead of training with a single batch, we train with multiple batches? So over here, we train with batch 1 and batch 2. And the autoencoder learns the projection into both training batches, including the batch effect. So this means that the batch effect is learned as well, which is not good. So that is why we always train with a single batch of data. Now, if we can only train with one batch of data, what if that single batch does not contain all cell types? So for example, now batch 2 contains a new type of cells, which are circled over here, called NK subset. These are slightly different from the regular NK cells over here. And these are absent from the training batch, which is still batch 1. So the autoencoder actually does not learn the difference between the regular NK cells and the NK subset. And what happens is the NK subset will become projected into the NK cells. In other words, cell types that are absent in the training batch may become merged into other cell types. And so now we have a problem. Because we can only train with one single batch, but there may not be a single batch that contains all the cell types we may encounter in the study. So how do we cover a diversity of cell types for training? So our solution is to use an ensemble of autoencoders. Now this ensemble contains multiple autoencoders, but each autoencoder is still trained with a single batch. So in this case, autoencoder 1 is trained with batch 1, autoencoder 2 trained with batch 2, and so on. Now, since each autoencoder is trained with a single batch, batch effect is not learned. But the ensemble still allows multiple training batches to cover all the diverse cell types in your study. So, putting everything together, here is an overview of MapBatch. First, an ensemble of autoencoders are trained, and each is trained with a single batch. So after training, you get your input data, which consists of a large number of batches. So all these are fed into the trained autoencoders. And the batch normalized data in low dimensional latent space can be extracted from the latent variables. And these can be used for latent level analysis, such as visualization, clustering, and so on. And batch normalized data in gene space can be extracted from the autoencoder's output layers. And these can be used for gene level analysis, such as differential expression. Okay, so here are some results. We created a benchmarking data set with simulated rare cell populations. So this is based on 6 batches of PBMC data, which are publicly available from 10x Genomics. We modified this data set to add rare cell populations. So in batches 1 and 2, we perturbed a small number of B cells to add a subpopulation of perturbed B cells that are only present in batches 1 and 2. Likewise, in batches 3 and 5, we perturbed a small number of monocytes. In batches 4 and 6, we perturbed a small number of T cells. So in this resulting data set, all 6 batches contain the usual PBMC cells, while the perturbed cell types are contained in only subsets of batches. So we tested MapBatch along with 3 other commonly used methods: Seurat, Harmony, and Liger. So actually, all methods can normalize batches really well. Batches become well mixed. However, only MapBatch maintains the perturbed cell populations after normalizing. So these perturbed populations are maintained as distinct clusters, and they are actually located near their parental cells. For the other methods, the perturbed populations were mixed into their parental cells. So this shows that MapBatch successfully keeps the biological signal necessary to maintain these rare cell populations. On the other hand, the other methods obscured them after normalizing. Another advantage of MapBatch is that it is extremely efficient in normalizing large amounts of data. So MapBatch's running time actually scales with the number of training batches used. And it takes about 2 minutes to train an autoencoder. Subsequently, after training, using these trained autoencoders to normalize large numbers of batches is extremely fast. So it takes about 3 seconds per batch. In real-world use, typically you only need to use a small number of batches for training, like 2 or 3 batches. So after this training process, you can easily process huge numbers of batches. So in my projects, I've normalized 100 batches of single RNA-seq data. Now, on the other hand, Seurat's running time scales linearly with the number of batches to normalize. So processing large numbers of batches becomes infeasible. Okay, so let me show you some real-world applications of MapBatch. So we did a study in multiple myeloma, and we used MapBatch to integrate and normalize 14 cancer tissue samples. After normalization, we did clustering, and then we identified a cell subpopulation called cluster 26, which is a very small population of cells found in only a subset of patients. And we found that patients with high numbers of these cluster 26 cells had a worse prognosis compared to patients with low numbers or no cluster 26 cells. Subsequently, we validated this finding in independent patient cohorts. I want to note that only using MapBatch will be able to find this cluster 26. Other methods that we used obscured this population of cells. Here's another project in colon cancer that we did. So we integrated and normalized 72 colon tissue samples, coming from a range of disease stages, normal tissues, polyps, and cancer. We identified cell subpopulations that were associated with disease progression. So in other words, from normal to polyp to cancer. We found a subset of fibroblasts that expands during disease progression. Another subset of fibroblasts shrinks during disease progression. Okay, so in conclusion, many batch normalization methods remove batch effect at the expense of biological signal. On the other hand, MapBatch retains more biological signal, and this enables us to discover rare cell populations. MapBatch is available as a Python package. It has tutorials, guides for all steps of data processing, the pre-processing, model training, and batch normalization. If you'd like to find out more, or if you're interested in trying out MapBatch, you can contact me at this email. This project was a collaboration between NUS, Proteona, and AI Singapore. And my professor was Professor Wong Lim Soon. Okay, thank you.
Thank you very much. Are there any other questions from the audience?
Yes. Okay, so each autoencoder is trained with a single sample, and there's a bunch of autoencoders. So multiple samples are used for training. But ultimately, all the other samples will be mapped into the biological space of these training samples. So yes, in the sense that you do assume and you hope that your training samples basically cover all the biology in your study. If it doesn't, then yeah, you will be missing out on some biological findings.
I have a question.
Yeah.
So your approach is currently being tested, examined using single-cell RNA sequencing data. Would it be generalizable to other different types of single-cell modality like proteomics as well?
Yeah, good question. Yeah, so currently we are using it just for RNA data.
Yeah.
But the principles behind it should apply to other omics data as well.
Yes.
So,
and also, like I suppose between different type of batches, there may be some missing data, like data imputation issues that you have to deal with. How that impact, the potential impact on the performance of your algorithm if there is a larger percentage of missing data.
Yeah, so in single cell data, there is the case where certain genes just don't show up in the analysis. Yeah, so unfortunately, those would just be missing from whatever findings you want to make.
Yeah. Thank you much.